Hi everyone. Today I will be telling you about my final project, which is on ultra-deep single-cell proteomics for understanding neural tube patterning. So as a preface, the neural tube is a structure in a developing human embryo. I think it develops during the third or fourth week of pregnancy, but it is the first part of the central nervous system in a human embryo. So it becomes what ultimately in a human, an adult human, is the spinal cord and the brain. So protein patterning through morphogen gradients is critical for this process of in spinal cord and neural development. So down here you can see like a diagram of a human developing spinal cord. And what you might notice is that there are these very significant gradients in proteins like RA, FGF and Wnt, SHH, TGF beta. And each of the concentration of each of these proteins, which is just basically what a morphogen is, it's a protein that is regulated by its concentration, is important for specifying the anterior, posterior, dorsal, and ventral axes. So the amount of each of these proteins is what specifies and dictates the fate of a cell in a lot of these progenitor cells. One of the most important gradients, as an example here, is SHH. So here you're looking at the same like notochord and spinal cord down here, but you're looking at a cross section of that. And so here they're isolating that SHH gradient. And you can see that the concentration of SHH is actually what dictates something called the neural tube closure during neurulation. And the closure of the neural tube is what eventually becomes your spinal cord. So before you have like a neural groove and then it closes, and then that tube becomes the spinal cord in the brain. And the closure of that neural tube is extremely important in human development. So if that neural tube is not closed properly, you can get something called a neural tube defect, which is a whole class of neurodevelopmental disorders. It's characterized by an incomplete closure of the neural tube in 3 separate areas, but there are several subcategories and subdiseases in this class of neurodevelopmental disorders that are not very well understood, especially because the closure of the neural tube is dictated by, like you saw in these past slides, very fine gradients in protein levels that happen at the single cell level. So here you would have a single cell that is very high in RA and very low in FGF and Wnt and very high in TGF beta, versus a cell a little bit to the right, which has slightly less levels of RA, slightly higher levels of FGF and Wnt, and so on. So at this point, we don't have a very good idea of the proteomic landscape of the neural tube. So previous work has applied proteomics for developmental biological problems. As an example, Gomez et al. Used LCMS and LCMS-MS to map the proteome and the phosphoproteome along the dorsoventral axis in Drosophila embryos. So here you can see they were able to achieve spatial resolution, and they use this to show dynamic genotype-dependent changes in phosphorylation of these of certain proteins. Other studies have actually done single-cell proteomics on blastomeres. They isolated individual blastomeres by dissection and did single-cell proteomics on these Xanapis blastomeres. And finally, most recently, we see a paper that actually got developing human brain tissue samples and did FACS and single-cell proteomics to get proteome level data in a developing human brain. Now, all these studies have been very interesting in laying the groundwork and showing the feasibility of using proteomics and even single-cell proteomics in developmental contexts, but no study at this point has performed both time-resolved and spatially resolved single-cell proteomics, and specifically during human neurulation and on the neural tube. So this is a gap my proposed workflow addresses. So to this end, we hope to perform an ultra-deep, label-free single-cell proteomics platform across 3 stages of neurulation in human amniotic fluid-derived iPSC spinal cord organoids, so that we can map the proteomic gradients that drive neural tube formation and eventually, hopefully, get some more insight into neural tube defects that are not very well characterized at this point. And so we hypothesize that there are fine-scale dorsal ventral and anterior posterior proteomic gradients that exist during neurulation, and that these gradients can be resolved at a single cell resolution using DIA-based label-free proteomics. And to this end, we will use SciProChip, which has been reported in literature previously, but it is a microfluidics platform that automates single-cell lysis, digestion, and cleanup in nanoliter level volumes, which is like the volumes that we'll be working with. And it dramatically reduces sample loss and enables deep and reproducible single-cell proteomics. So figure A is like literally a picture of the chip. And then in figure F, you can see all the things that the chip is doing, like trapping the cells, counting the cells, lysing, digesting the proteins, desalting, and preparing a sample that is ready to perform mass spec on. And so we will be obtaining these samples and these single cells from human amniotic fluid derived iPSC spinal cord organoids, which are basically iPSCs that come from collected amniotic fluids from ongoing pregnancies that are reprogrammed into spinal cord organoids. And in this way, we can replicate the exact genetic background and developmental context of a neural tube and the folding of the neural tube, while remaining non-invasive and lowering the cost, as well as avoiding the issues associated with like the other study I mentioned, which actually got like real human embryonic brain tissue and performed single cell proteomics on that. And then so we can also even scale the platform. We will also endogenously tag proteins that characterize each part of the axis with a fluorescent reporter so that we can perform FACS and isolate single cells at a spatial level. And we will do the same process at 3 different time points in the neurulation process, so that we can achieve both spatial and temporal resolution. And so as an example, we could use PAX7, PAX2, and OLIG2, which are very specific to different parts of the embryo. And this figure is just an example of what that might look like. In order to achieve the single cell level proteomics and actually get some viable data, we first will achieve ultra deep resolution by cleaving our proteins with multiple orthogonal proteases. So we'll aliquot parts of the sample and cleave them with different proteases so we can avoid like structural constraints, as well as achieve like a more comprehensive and holistic picture of a fragmented protein, and avoid the costs and avoid the implications of like using a single protease, which might be sterically hindered because of the structure of a protein. We'll also use ultra low flow nano LC with very low flow rates to sharpen our peaks and increase the separation efficiency and resolution of our chromatogram. Much like the ultra deep proteomics paper we read, we will also be fragmenting our peptides with multiple techniques like HCD, CID, and ETD for high sequence coverage. And so using all three methods increases overall peptide identification, because we can identify both a B, a Y, and Y series and a C and Z series through these different fragmentation techniques. And so HTD and CID, for example, are collision-based, and ETD is electron transfer-based. We will also be using a data-independent acquisition method, which, as we've talked about in class, is the process of isolating all precursor ions into a predefined isolation window and fragmenting every single precursor ion, as opposed to DDA, or data-dependent acquisition, which only fragments like the high abundance peptides. And so in this way, we can achieve single cell proteomics of like discovery proteomics and like even quantify low abundance proteins. And then we'll use a DIA-NN, which reduces noise, and we'll be able to score and match our DIA spectra to predicted protein levels and protein characterizations. So ultimately, we anticipate developing a pilot atlas that identifies proteomic signatures and disrupted morphogen gradients that cause neural tube defects, and provide a molecular and mechanistic basis for previously unexplained cases of these diseases. In future works, we can expand this platform to different PTMs. We can also study other developmental biological workflows. And finally, we can even potentially identify biomarkers that can be used to detect these defects earlier on in pregnancy and using less invasive and like high cost methods. So thank you all for listening. Thank you for a great semester.